I can also target proteins in here. What does that mean? I can use a dye that I can hook on to an immune complex. You've heard of immunity, right? We're all trying to get immunity with COVID, right? Hopefully. Herd immunity, you've heard that term, right? So immunity is an amazing system in your body because when you get infected by an antigen, your body makes antibodies. You've heard of antibodies, right? And they're like Legos. They hook on to each other. So I can take advantage of that hooking. I can put a dye on there, and I can tell this molecule, go find me actin, or go find me RNA. And that's exactly what I did with this cell. I stained this with acridine orange, which goes and finds RNA. Now look at it under fluorescence. Look at all the RNA in this cell. That shouldn't be there. These are impossible pictures. So we're finding at the molecular level some of these proteins that shouldn't even be there just in the osteocytes. Now I can also stain them with a vital stain, and that's a stain that you can use while they're alive. So this is an interesting test. If I were to grow some of my cells in a petri dish and stain it with a vital stain, it would take up that stain. It's behaving as a normal tissue. Now these guys are supposedly 67 million years old, but they're taking up stain. It's taking up a vital stain, toluidine blue. So there's all kinds of stains that I can test with in the laboratory, and every single test we've done has shown these are behaving like normal tissues. So we've got normal tissues. And here's an example of some cells that don't take up the stain. See, it's purple here and not here. And why is that? Some of these cells don't take up the stain. Well, all of your cells are coated in a layer of fat. Okay? It's called the phospholipid bilayer. Your cells have a layer of fat around them. And over time, there's this thing that forms called grave wax. If you look in the forensic journals, the journals that study the decay of living systems, we get grave wax. And what that is, the wax just kind of hardens and concretes on itself over time. That makes an impenetrable barrier in the cell so the stain can't get in. So we see selective staining sometimes, but a lot of the cells do take up the stain. Here's a perfect example. Look at that cell that took up that purple stain. So it's behaving like regular tissues. All right, let's go back to the biofilm argument. Remember that biofilm that the bacteria make when they grow in population, they coat themselves in a glue. And this is a picture of a biofilm, okay? Inside the biofilm is mostly this milky substance, and then you see the cells. These are all microorganisms in here, okay, that are growing, and they all took up the stain. But the biofilm doesn't take up the stain. Do you see that? It's only the cells that stain in the biofilm. If this was a biofilm, it wouldn't take up the stain. But it's taking up the stain. So it's behaving as a real tissue. So they don't behave as biofilms, only the microorganisms. And here you see them here. This is a scanning electron microscope picture. Here are all the cells. And this is the biofilm. Look at that stretchy, gooey stuff. This does not take up stain. And so if those cells were biofilms, they would not behave as they are behaving.